Okay, so starting from where we left in the last class, last lecture, that because of unequal distribution of different kinds of mRNA in a cell, so if you are trying to make a cDNA library, there will be a skewed population of members of cDNA in your collection. If you want to view it pictorially, imagine this is a bacterial plate. You do not know which bacteria harbors what plasmid, because all you have done is you have taken your cDNA, like you have taken your mRNA, reverse transcribed it, then PCR amplified it, cloned it in a vector, and then you have transformed the whole mixture into bacteria, and then you have plated the bacteria, and you can see a few colonies. Just by looking at the colony, you cannot tell which colony harbors what particular circular plasmid. I am presenting a hypothetical scenario. If you sequenced, you will perhaps see something like this, that there are many A's and B's and some C's and maybe just one D and one E. Okay, these are different genes where A and B are perhaps your actin and tubulin. C is perhaps an enzyme in the glycolytic pathway, while D might be a transcription factor and C might be a ligand for a signal transduction pathway. So there will be this kind of very extreme varied distribution in a cDNA library. This is just for you to remember that whenever you are dealing with cDNA library, even now people use it. So there you will have this kind of skewed bias. Okay. Anyway, so now today I will be particularly talking about DNA sequencing. Okay. In DNA sequencing, I will take you to the history. First, I will talk about Sanger's sequencing, and then I will talk about what is referred to as next generation sequencing. I will discuss what are the differences between the two platforms and why this next generation sequencing is a talk of the town. So before we get into Sanger sequencing, first thing is you need to understand that there is something called DNA gel electrophoresis, which is not a very complicated concept. You form a gel. If you do not want to resolve the DNA based on sizes too fine, then you can make a gel with agarose, which is routinely done in the laboratory whenever you are cloning a DNA or doing a PCR amplification. You use an agarose gel, and you vary the percentage of the agarose gel from 0.8 to 2.5, sometimes 3%, to achieve higher and higher resolution. The more the percentage of agarose in the gel, more difficult it is for the DNA to move through it. Therefore, the resolution, meaning the difference in migration between a larger and a smaller DNA fragment, will be more, but I have to be very, very careful here that this higher percentage is particularly used if you are trying to resolve relatively smaller fragments which are varying in length maybe by 25-30 nucleotides or less than that. Okay, but anyway, but if you are trying to resolve DNA fragments that is varying in size by one nucleotide, only one nucleotide, okay, then you need to use a polyacrylamide gel. Okay? It's the same kind of gel that is used for protein, but polyacrylamide gel is also used for separating DNA. But typically the fragment size will range between 30 to 300 bases, and but the resolution is even if they are varying in size by one nucleotide, you will be able to tell. Okay, so and the DNA moves within this gel because you apply electricity. DNA, as you know, because of its phosphate backbone, is negatively charged. So if you put a positive end at one end and negative end on the other end, DNA will migrate from the negative end towards the positive end, and depending on how small it will migrate faster. Okay, so that's all. Now, how was I'll come to Sanger sequencing in a minute, but before that, let me tell you a couple of properties of DNA synthesis, which is critical for our understanding of both PCR reaction, polymerase chain reaction, as well as for DNA sequencing. So the first point that I'm sure all of you know, that DNA extension always happens in the 5' to 3' direction. The 5' to 3', how do you know? Because in the 5', the base will have a phosphate group at the 5' position of the sugar, and in the 3' end, the sugar will have a hydroxyl group in the 3' position of the sugar. Okay? So that's how it is. The next nucleotide will be, so if this is DNA, then the next nucleotide that will be added to it, let us say that it is colored in green, so that will be added here. Okay, then the next nucleotide that will be added to it will be added here. So this is the 5' end of the DNA, this is the 3' end of the DNA. So DNA elongation is always from 5' to 3' end. Number two, this is very important. I apologize for my handwriting. Please search this word in the dictionary. It's called de novo. DNA can never be synthesized de novo. Please remember this always. What it means is that if you take 2-deoxynucleotide and use a DNA polymerase, then DNA polymerase will not be able to polymerize them and make a 2-nucleotide long DNA strand. Okay, DNA can only be extended. Okay, so DNA synthesis by a DNA polymerase can only happen as elongation of an existing DNA. DNA synthesis can never happen de novo. Okay, so this is a very, very important property for all of us to remember. This is how it differs from RNA synthesis. RNA synthesis can happen de novo. Okay, DNA synthesis, no. And the other thing is that DNA synthesis is always by DNA polymerase. You know, there are some enzymes that can do chemical synthesis of DNA. We are not getting there. DNA synthesis by a DNA polymerase is always template driven. Okay, so you will always need a template. Okay, so what it means then that that if you have a DNA, this is 5' to 3', and you make another DNA that is 5' to 3', and then you add a polymerase, then one base will be added to it. Then the polymerase will add the second base to it. Then the polymerase will add the third base to it, and so on and so forth. Eventually, this strand will be elongated and it will end here, and you will make a antiparallel double-stranded DNA. Now, what happens in polymerase chain reaction or PCR reaction? Again, I would like to tell you about the history. This is another of my very favorite examples to convey that groundbreaking work that has enormous commercial and societal importance did not start as a process of a commercializable research. It started with a very simple fundamental query. The query was that there are these places in the US called, there is this place in the US called Yellowstone National Park, where there are some hot springs where the temperature is around 90-95 degrees centigrade, and it has very high sulfur content and all sorts of things. pH ranges anywhere between 1 to 10, depending on which hot spring. But there are life forms in there. There are some bacteria that live in there. So, but your body, my body, many of the other organisms, they typically operate at 37 degrees centigrade. So therefore, the scientists, and any of the enzymes in our body, chances are that if you heat it up to 90 degrees centigrade, the enzyme will denature and it will not be able to function. But the fact that these bacteria are living in the Yellowstone National Park geysers or hot springs indicates that all the metabolic processes are happening there, including DNA synthesis, right? So chances are that the enzyme that is synthesizing DNA for the bacteria living in these hot springs, they will be temperature stable. So there were a group of scientists that simply wanted to know that what are the biochemical characteristics of molecules that are there in these bacteria. And that's how they identified the DNA polymerase of now famous the Thermophilus aquaticus, or which you know as Taq. This enzyme remains active even if you heat it at 95 degrees centigrade. It does not polymerize at 95 degrees. It typically polymerizes around 73, 74 degrees centigrade, but it remains active at 95 degrees centigrade. And this became enormously beneficial from a biotechnology application perspective, because before this, the enzyme that was used for DNA synthesis, a primer-dependent DNA synthesis, is clenopolymerase. Clenopolymerase is a fragment of E. coli DNA polymerase that could do the DNA synthesis and some proofreading action as well. I'm not going to get in there. But the problem was that, you will take a double-stranded DNA. Okay, you will take a double-stranded. Oops, sorry. You will take a double-stranded DNA. You will take a double-stranded DNA, then you denature them. Okay, so you will get a red strand, and you will get a green strand, and then you will use primers, and by that time people knew that the DNA sequences, the double strand is complementary to each other, they're anti-parallel, so they knew what to do. So if they knew that this is um, uh, this is sorry, it's not working well any longer. Yeah, if they knew this is 5 prime to 3 prime, and this is 5 prime to 3 prime, they will design primers such that this will be 5 prime to okay. So imagine this is 5 prime. So this is 5 prime to 3 prime, and then it will elongate like this. And here they will design, like the other strand, which will be 5 prime to 3 prime, and then that will elongate. And then you have all the DNA strands that you had originally, you denatured them, and they all became double-stranded. Now you cannot do another round of amplification, okay? What you have to do is then you have to denature all of it. In the process, the clino enzyme will get inactivated, and then you add fresh round of primers and enzyme, and then you get the next round of elongation. So that would have been, that was very, very time consuming. But with the advent of Taq DNA polymerase, this process of denaturation, another round of primer ligation, primer annealing, and elongation can be done in a sequential manner without any need to stop the reaction or stop the reaction or manual intervention. So then this whole process was automated, like machines were developed where the temperature can cycle between 70 and 95 degrees centigrade, and then you can get rapid amplification of DNA, which is polymerase chain reaction or PCR reaction. In the context of understanding the genome, if you knew the sequence of any part of the genome, no matter how you know it, you can design 2 primers. They have to be reasonably close, 1 kb, 2 kb, 3 kb at max. They have to be reasonably close to each other. And then if the 2 primers that you design, if this is the genomic DNA, double-stranded, then you will design 1 primer from here. Let us say this is 5 prime, this is 3 prime. Please never write DNA or RNA without 5 prime, 3 prime. So this will be 5 prime, this will be 3 prime. It is very important. Otherwise, like you will draw primers that will move away from each other. So always write your 5 prime and 3 prime. Okay? And it is very simple because you know that they will have to be anti-parallel. So very simple. So that way, this will elongate the first round. This will elongate and it will go beyond. It will go beyond. But in the second round, here the primer will anneal only here. Okay, and this primer will anneal here. So there you will get the defined length. So you can amplify any segment of the genomic DNA by just careful use of primers and PCR amplification, to generate any length of genomic DNA, and then you can clone it. Okay, and the utility of that we will come to it very soon. Let me just wipe off all these things. Okay, so yeah. So that's what is PCR reaction for you, and we have already talked about reverse transcription PCR. What you do there is you take your oligo DT and which will anneal to the poly A tail, and you do it. I'm not going to talk about it again. You can sequence different parts of the genome if you have some inkling about the length that you are trying to sequence by cutting it with restriction enzymes, then ligating it to make it a circle, and then doing PCR. But this time, normally what you do, please note carefully, normally what you do is your double-stranded DNA will be like this, and your primer will be like this. Okay. Okay. See, I made a mistake here. Okay, so that's what 552 3 prime, and then the primers are facing towards each other, so the elongation will be happening towards each other. But in this particular case, you see the primers are facing away from each other. So here the elongation will happen this direction. Here the elongation will happen this direction. But that way you will get the entire sequence amplified, and then you can like the entire length amplified, and then you can sequence it. So if you knew sequence of only this part, you will be able to know the sequence of this unknown part by this method. Like these are all different methods by which genomic DNA was sequenced in the olden days. Now those are all irrelevant methods, but you should know how science progressed over time. Now let me come to DNA sequencing. Okay, so before I before I get to this method, I should tell you how. And of course, you know that this method was developed by Frederick Sanger. He got the Nobel Prize for developing it. So first, let me tell you how the DNA sequencing was done long time back. When I used it, I was perhaps at that juncture. I have seen two revolutions in terms of DNA sequencing, and I will elaborate both. So when I was doing my PhD, again, if you look at my first paper, I cloned a gene. Okay? When I say I cloned a gene, it does not mean what you mean today, is that you have a piece of DNA in one vector and you cut and put it in another vector. That's not what it is. When I say I cloned a gene, meaning I discovered the sequence of a particular gene and the function of that protein. Okay, so that was the first time that protein sequence and the gene sequence was discovered. That's what we meant cloning a gene. And in those days, how we did sequencing using Sanger's method is like this. Say, let us talk about the same sequence. Let's say this is the sequence. Okay, TAC, GC, GTT, whatever. Okay. Now you will have 4 different tubes. 1, 2, 3, 4. 4 different tubes. I don't know why in certain places it does not work. Okay, 4 different tubes. Okay. In each of these tubes, you will have a radioactive deoxynucleotide. Okay, typically it would be radioactive C, cytosine, so that you can easily determine the size of the DNA. Radioactivity was only for that purpose, nothing else. And in each of these tubes, you will add a dideoxy variant of the nucleotide. Okay. So in this one, it will be dideoxy A. In this one, it will be dideoxy T. In this one, it will be dideoxy C. And in this one, it will be dideoxy G. And you will add a primer because you have some idea about the DNA, or if it is cloned in a cloning vector, so you will use, remember those T7 or T3 promoter sequence or SP6 and 13 forward reverse. Those were the primers. So if you use any of those primers, you can use that. Then you add cleanoplot polymerase, which is the DNA polymerase, and you add these dideoxynucleotides and radioactivity. These dideoxynucleotides will be very tiny fraction of the total nucleotide concentration, typically 5% of the total nucleotide concentration. Therefore, every place where an A has to be incorporated, generally speaking, DA will be incorporated. But occasionally, okay, a DDA or dideoxy A will be incorporated. And wherever it is incorporated, that chain will terminate right there. So I'm just giving you an example from what will come out of this tube. So what will come out of this tube will be, let us say that this is your SP6 primer. Okay. So then in some you will have only this much of elongation, in some you will have this much of elongation, in some you will have this much of elongation, and or this much of elongation, and things like that. Please note that in all the tubes you have the same primer. Now, because it is a DDA, so the first one that will stop here will be right here. The next one will stop right here. And the third one will stop right here. So you will generate varying fragment sizes only depending on where the next A is. Similarly, you will generate different sizes from this tube, depending on where the T's are, different sizes from this tube, depending on where the different C's are, so on and so forth. And you will run them in 4 lengths. Okay, so your gel will have a T len, a A len, a G len, a C len, and a molecular marker. Okay, so the marker will have a resolution of 1 base. Okay, so you will have exactly the length of the fragment, and then some will stop here, some will stop here, then some will stop, like depending on what is the size. They will all stop at different places, and you read the sequence from the bottom. Okay, so that way we used to do our like the DNA sequencing during our time. So for each fragment of DNA, you will have to do 4 different reactions. Okay, so that was then. Then somewhere around 2000, when fluorescent technologies started to develop quite rapidly, then the scene changed a little bit. How did it change? That instead of this radioactive nucleotides, people started using fluorescently labeled nucleotides, and the fluorescence level would typically come with a color code. Like I still remember that A would be in blue, G would be in green, T would be in black, and C would be in yellow, something like that. Okay? So these are 4 different kinds of fluorescence. Now, you will have these dideoxy ones, which are all fluorescently labeled. Okay, and wherever they are incorporated by chance, that's where the DNA elongation will stop. Okay, so a fluorescent nucleotide gets incorporated and DNA elongation will stop right there. Okay, and but you do not need to do any longer 4 different tubes. You can all do in one single tube, because you will know where the reaction stopped based on the color of the product, because the color will come from the fluorescent fluorophore, and the fluorophore is only with the dideoxynucleotide. So wherever a dideoxy has been incorporated, that's where it will stop, and that's what will give you the color. So from 4 tubes, you have now come to 1 tube, but still you can sequence only 1 template in 1 tube. Okay? And you will get this color. Okay, so the color coding is shown here. You see, G's are in black, C is in blue, T is in red, and A is in green. Okay, so that's how you get to do the sequencing. Okay, closest to the primer, if it stops, that will be the smallest product. Then the next one, then the next one. So here it is only showing you the products because of stoppage at T, and similarly you will have products because of stoppage at A, or because of stoppage at C, or because of stoppage at G, and so on and so forth. But now you can do all in 1 tube, and you simply run it on the gel, and because you have single base resolution, so you can read from here how it is reading like. Okay, so that's how it is. It is done. Even now it is done like that. Even now, Sanger sequencing is the mainstay for most molecular biology laboratories, including my laboratory. But this way of sequencing came as a major hurdle when we wanted to sequence the entire genome, because the entire genome, you know, like if you were talking about human genome, it is 6 into 10 to the power 9 bases per cell. And so the genome is 3 into 10 to the power 9 base pair long. Now, if you can, with Sanger sequencing at best, per reaction, you can get 1000 nucleotide sequence. So that means that you will have to do 30 million sequencing reactions to cover the entire genome, and that is a whole lot of time. And you know, like there are many logistical hurdles in there, so that makes it very difficult. Though that you should know that that is how the human genome was sequenced. Okay, human genome sequencing that was published for the first time was done using Sanger method only. Like Craig Venter introduced a very clever technique that I will come to in the next lecture, but it was primarily done with Sanger sequencing. However, soon after genome sequencing was accomplished, like I still remember, around 2004, 2005, a new method of DNA sequencing started to make news, which is called the next generation sequencing. So what is the fundamental difference between the Sanger method of sequencing and next generation sequencing is this. In Sanger method of sequencing, in a particular reaction tube, you can sequence only 1 template. If by mistake, you had 2 different templates in the same tube, like say you mixed the plasmid from 2 different bacterium in the same tube, then your Sanger sequence will go completely, you know, wrong, because remember the primer will bind to both the plasmids, but the next base that it will face, instead of this T, it might be an A here, instead of this A, it might be a C here. So you will get all sorts of extra peaks here, and it will be all jumbled up. But in next generation sequencing, we now have the ability to sequence billions of fragments simultaneously in the same reaction mix. The way it is achieved is by partitioning the reaction space into very tiny but precise spaces, and you can read the sequencing as it progresses within each of these tiny spaces. It might be a little difficult for you to imagine what I'm talking about. I will try, but you have to read up yourself as well. Imagine this is a matrix, okay? This is space under observation. And you have somehow named them, okay, as different zones, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, like this. So next generation sequencing allows you to sequence a particular DNA at position 1, another DNA at position 2, another DNA at position 3, another DNA at position 4. This way, billions of DNAs in this kind of very small, tiny spaces that are, separated, not necessarily physically, but partly chemically and partly through image analysis. And I will explain how it works. Okay, so how does it work? So what you do is you take the genome, okay, and then you fragment it. Okay, you cut it into small pieces. Okay, and you can use a tetra cutter to get small pieces of DNA, and then you use like this two adapters. These are known sequences. These sequences, and you can add them to a small fragment of double-stranded DNA. So if this is your double-stranded DNA, you add adapters right after this, okay, right, like this, okay, and like this. So you can, like these are defined sequences. These sequences, okay. You just make use of ligase enzyme, and you add them, and these are the same adapters. So every piece of genomic DNA fragment that you have generated will be decorated with these adapters on either end, and the sequence of these adapters, and then you denature them. Okay. Then you denature them, and you have a glass slide. You have a glass slide which has this kind of poles, spikes. These are small DNA sequences that are complementary to this adapter. Okay? Because the adapter sequence, you do not know this sequence. You have no idea what this is, but the adapter sequence. So you can have, like, you must have read, or if not, please read up about solid phase DNA synthesis. So this is, if you think of inkjet printing technology, when you print something on a printer and use an inkjet printer, just imagine the process of printing a fantastic photograph. Okay? Different places of the photograph, the frame, will have different kinds of colors, and these are not true colors, these are mixture of colors. So particular ratio of colors or inks, and you have only 3 kinds of inks. Remember, you have red, green, blue, and black. Only that's all that you have, right? So a computer program decides for each of the tiny spots in what ratio these inks will be mixed to accurately reproduce the image that has been captured on the camera. Okay, or like. So therefore, there is a technology already existing to precisely deposit an ink on a solid surface. This is called inkjet printing technology. And the way of attaching something to the glass covalently is a technology called lithography. So if you combine these two and you deposit precisely at different spots a mixture of enzyme and nucleotide and other reagents, and you do it in repeated manner. So in every spot on the glass, you can dictate a particular length of DNA to be synthesized there, and they will, my drawing skills are really bad, so they will pretty much look like little spikes coming out of the slide, and the sequence of this. Okay? So you will allow this denatured DNA with this adapter to anneal to those, and then you use PCR reaction to amplify this DNA with adapter bound on both ends within a very small region. This is what is shown, like a 10x PCR, you have a few thousand identical pieces of DNA clustered in one, and in the actual glass slide you will have billions of these clusters, or at least millions of these clusters. Okay, each of these cluster will have few thousand copies of the identical DNA, but the cluster that is here will be different from the cluster that is here, will be different from the cluster that is there, and so on and so forth. Okay, so each of the clusters is few thousand copies of the same piece of DNA, flanked by adapters for which the sequence you know. Okay, so after you have done this and you have done some other tricks to linearize them, now what you can do is you can flood this system with this time there is no dideoxy. Okay, you will not stop the reaction. There is a proprietary technology, at least I do not know what the technology is, but I will tell you what it does. So you will throw in these 4 fluorescently tagged nucleotides and add polymerase, and then this will be added to the primer, but the addition will be based on what is there on the other strand, right? Because DNA polymerase can only synthesize DNA based on a template. Okay, so the beauty is that the moment the first nucleotide is attached, it is such is its chemical nature that it does not allow addition of the second nucleotide. But the nucleotide that will be attached will or may be different in different spots. Like if we call these 1,000 fragments as one spot, so it will be different in different spots. So if you follow this particular spot here, then you see that in the first reaction it incorporated blue. That means it was A. Then you have to do a chemical treatment. This is all done robotically nowadays. You do a chemical treatment, the fluorescent tag is removed, and once the fluorescent tag is removed, then it is ready to accept the next nucleotide. And you can see that the next nucleotide that is added here within this circle that you can see is red in color, so that is C. Then again you do chemical removal of the tag, it is allowed to bring in the next one, another C. Then a T, see the green color? A T. And then, so you actually image it using a modified microscope, because, like you were talking about, like a tiny glass slide, half an inch by 2 inch. Within that you have a few million spots, and within each spot you have cluster of thousands of DNA that are identical. Okay, but because of this 1,000, the fluorophore incorporation is 1,000 times. That's why that's where the intensity comes from, the sensitivity of the next generation sequencing comes from. Okay, so you will be able to scan, like after incorporation, you scan, image, chemical treatment, remove the fluorophore, then you add another round of fluorescently labeled nucleotides, again scan, image, remove the fluorophore chemically, and the next round you can keep on doing, and for every frame you have an image. So, and there are very sophisticated computer algorithms, image analysis algorithms that can track each of the spots, and that will tell you that how the nucleotide is changing over in a particular spot, and then you will know, okay, so this is the sequence of spot number 1. Okay, typically you will have a grid. Okay, so of course, I cannot draw a grid for million spots here, but here you will have A, B, C, D, like that. Here you will have 1, 2, 3, 4, 5, 6, like that. So you will have a unique grid position. If it is A4, it only means here. If it is D13, it only means here. So you will be able to track through image analysis how the sequence is changing at each of these unique spots. So you will know the sequence at each of these unique spots, and you will know that for million spots simultaneously. So this is what is referred to as next generation DNA sequencing. There are different formats. One of the most popular ones that you have read, in popular literature and other places is Illumina sequencing. Then there is another one from Roche that is kind of a variant of this. But there is another one from, it's called ion torrent technology from Light Technologies, which is slightly different. Okay, the way it is different is that in ion torrent technology, you do the same thing. You have an adapter, and you have the actual DNA fragment, covalently linked to this because of the adapter, and then you have the other primer, and then you start the sequencing, the elongation. But in this case, what you do is slightly different, that you have this matrix. You first flood the whole thing with a chemically modified variant of any one nucleotide, say A. Okay, and wherever A will be incorporated, there will be a small, electric signal there. Electric signal. This time it is not fluorescent signal, it is electric signal. That electric signal is generated because of the incorporation of the base. Okay, so you will have the information again on the grid in every grid location where an A is incorporated. It will not necessarily be incorporated in every grid location, right? Because every grid has a different DNA fragment, and you have a million different DNA fragments. And then you wash off the whole. Okay. Then you go in with a chemically modified version of T. Okay. And then you again locate what are the places where the T is incorporated, like where the electric signal was generated. And then you wash up the whole thing, then you come in with C. And you keep on doing this, and you keep on mapping where there was an incorporation event. The incorporation event is marked by a fluctuation in electric signal. Okay? Here it is showing as pH change, it is basically converted into an electrical signal. And this way also you can do the whole sequencing. This is called ion torrent technology. But nowadays, Illumina pretty much is dominating the entire commercial scene. But so two fundamental differences. One difference is that millions of reactions are being observed simultaneously. This is dramatically different from the Sanger sequencing. And the reason it is possible is, one, because of improvement in the lithography technique and inkjet printing technology and high-end imaging and image analysis. So you see 4 different kinds of technologies coming together on one platform to give you this very powerful technology. And there is another fundamental difference. That's what actually enables next generation sequencing, all methods. Sanger method is dependent on termination of DNA elongation. These methods, all of these, are dependent on addition of a base, elongation of a DNA primer. So that's how these two, the Sanger method and the new next generation DNA sequencing method are different. Like this is truly a disruptive technology, and now getting your whole genome sequenced costs only $1,000 or a little bit less than that, and it will take 3-4 days or at max. Compare that with when the first human genome was sequenced. I don't remember exactly, it was like 4 or 5 years long to get one human genome sequenced, and the cost was few billion dollars. Okay, so we have reduced the cost from few $billion to $1,000, reduced the time from 4 or 5 years to 3-4 days, and that is disruptive technology, that is disruption. Okay, and so next generation sequencing has completely changed the way biological science investigations are done today. I will elaborate that in the other lecture. Okay, so with that I will stop for today for this lecture, lecture 8. So lecture 8, your take-home message is primarily how the sequencing technologies evolved, and about PCR and RT-PCR and how they are critically dependent on the properties of DNA polymerase. The DNA polymerase, the key property being that it is always template-based elongation, and DNA polymerase can only elongate. No de novo synthesis of DNA is possible. Okay, that's all. Thank you very much.